Hello, we continue chapter 21, Immune System, Part 2. Here's your table of contents. And let's talk about origins and development of lymphocytes. We have two types of lymphocytes, B and T cells, and they are very important cells of our immune system. Both of them are born in the red bone marrow. Can you remember where exactly red bone marrow is located? Yeah, it's obvious in the bones, but which bones? Those would be most of flat bones, let's say our pelvic bones or our sternum, and some amount of bone marrow is located in epiphysis ends of large, long bones. After T and B cells are born, they mature in the primary lymphatic organs. B cells stay in the bone marrow, and T cells go through blood. They go to thymus where they mature. So thymus and bone marrow would be primary lymphatic organs. It takes some time for the cells to mature, but finally, when they are ready, they go to secondary lymphatic organs and tissues. And that's where they do their job. They interact with each other. They are looking for antigen-presenting cells and antigens to produce the immune response to fight antigens whenever it is necessary. And secondary lymphatic organs would be diffuse lymphatic tissues, lymphatic nodules, tonsils, lymph nodes, and spleen. So, while maturing in thymus or bone marrow, lymphocytes go through positive and negative selection. Remember, they should be able to fight pathogens, bacteria, and viruses, and cancer cells. So the process, which is called positive selection, tests those B and T cells, making sure that only those that react against antigens, fight them, will stay alive and later proliferate and form clones. Those lymphocytes that cannot fight antigens will be destroyed. So that's positive selection. Let's choose those that can fight antigens. Okay, that process is done. Now all the lymphocytes should go through another process called negative selection. Yes, they should fight, but they should not react against self-antigens. Remember, antigens are tiny structures on the surface of all the cells, including our own cells. And our own T and B cells are not supposed to attack our own body cells. So tolerance is a state of unresponsiveness of lymphocytes to a specific antigen, usually to self-antigens, our own cells. That's what is tolerance, please know that. So all the lymphocytes that try to attack our own cells will be destroyed in bone marrow or thymus, and that is called negative selection. Sometimes when something is going wrong with the process of negative selection and lymphocytes gain the ability to attack our own cells, that would be autoimmune diseases. Let's talk about activation of lymphocytes right now. So, when our lymphocytes are traveling through the blood and they are in the tissues, when they see bacteria or virus, they should be able to recognize those foreign antigens on the surface of bacteria or virus. What they do after they recognize the antigens, they must increase in number so they could effectively destroy all of those antigens, all of those bacteria, all of those viruses. And to be activated, to do that, to produce clones, they should be activated. So how do we activate our lymphocytes to fight some specific bacteria or some specific virus? Let's talk about structure of antigens first of all. So that is some foreign body bacteria or virus, and on their surface there are antigens. You know that. And on the top of antigens, there are what is called antigenic determinants. They have different shape, as you can see. And so here they are. Antigenic determinants are specific regions of a given antigen. Our lymphocytes on their surface have special receptors. Here it is, antigenic receptors on T cells and on B cells. They are sites of lymphocytes that can combine directly with antigenic determinants. So that lymphocyte is floating somewhere in blood plasma. It means a bacteria, and it tries to feed its receptor to that antigenic determinant of bacteria. If it fits, okay, now they know it's an enemy, it should be destroyed, and then lymphocytes will be activated. Activation also involves glycoproteins, a special type of proteins called major histocompatibility complex, or MHC molecules that are attached to cells' plasma membranes. What are we talking about? So what is major histocompatibility complex? Again, it is something attached to the cells. Type 1 or class 1 of MHC would be found on surface of our own body cells affected by pathogens. What does it mean? Our cell is attacked by the virus. Our cell processes virus, pinches pieces from it, and produces that MHC type 1. When cell is displaying that part of foreign antigen on its surface, it's like saying, kill me and kill that virus inside of me. So both that molecule that cell produces, MHC, and a part of that foreign antigen will be displayed on the surface of the cell. To understand it better, let's talk about it in details. That is one of our body cells, and it was affected by a virus. So that is some foreign protein, some virus, and our cell immediately starts attacking it and destroying it. It breaks it into the fragments. Each of those fragments is still a foreign antigen. Next one, in rough endoplasmic reticulum, a piece of that antigen will be connected with our own molecule. Our own molecule will be that MHC class 1, that major histocompatibility complex, and it will be attached to the particle, to that fragment of foreign antigen. Now our cells, through Golgi apparatus, sets that MHC together with foreign antigen particle on its surface. And now lymphocytes, other cells of immune system can see it. It's like a signal that cell is giving to all immune system. Destroy me, kill me, I have a virus inside of me. So it stimulates cell destruction. Let's summarize. So major histocompatibility complex class 1 is formed by the cell attacked by virus. That cell presents MHC together with foreign antigen on its surface, and it's asking to be destroyed. The purpose to destroy that virus inside of it. Now let's talk about major histocompatibility complex class 2. So again, we have particles of antigens presented on the surface of the cells, but now those are not regular body cells, those would be special cells fighting pathogens. So when antigen is presented on the surface of a cell using MHC type 2, those cells would be B cells, macrophages, monocytes, dendritic cells. So not your regular body cells, but special cells that's supposed to fight pathogens. And when those cells carry that foreign antigen attached to their major histocompatibility complex, it's like they show to all other immune system cells, there is an enemy, let's go and destroy it. That's how it works. That's one of our immune system cells. Let's say it's a macrophage. That macrophage takes inside a foreign antigen virus, exactly like antibody cell would do. It tries to fight it, tries to destroy it, and then it presents particles of that virus on its surface using that molecule MHC class II. Presented at it on its surface stimulates all other immune system cells. That is your MHC class II. Now about co-stimulation. So our B or T cells, when they meet any cell with MHC, they are not activated yet. They need to bind to that cell, and they also require what is called co-stimulation. So co-stimulation is a second signal that is needed by our T or B cells in order for them to produce an immune response. And it can happen by two ways: by cytokines, special chemicals released by the macrophage, and or by surface molecules. What it means? Okay, that is your macrophage. That macrophage took inside a pathogen before and presented it on its surface using MHC class II. That is your antigen. Macrophage met helper T cell, and macrophage wants to activate helper T cell. That connection that happens between MHC and receptor of T cell, that's the first signal, but we need a second signal, which is co-stimulation. Macrophage either produces cytokines, special chemicals that help to activate T cell, or macrophage makes a second connection through special surface molecules between itself and helper T cells. And only now, after that double stimulation, helper T cell or B cell will be activated. Before you continue, please stop the video, and answer the questions for chapter 21, part 1.1. Let's go to part 2.2 and make some summary about how our B cells and T cells work. So they were born, they were raised in and matured in B cells, T cells. They went through positive and negative selection, and now they are ready to work. What happens with B cells? After they are activated, they become either plasma cells or memory B cells. Plasma cells start fighting infection immediately. They produce antibodies, and those antibodies will directly or indirectly responsible for destroying antigens. So those are soldiers, plasma cells. They fight infection right now. Memory B cells, as well as memory T cells, will remember that antigen, and next time when we meet the same infection, they will produce a very fast immune response. So what does it mean? If somebody had a chicken pox when they were 3 years old, and now they are 10 years old, and they encountered that virus, chickenpox virus again, will they get sick? No, because as soon as that virus enters their system, their memory B cells and T cells, they remember the chickenpox virus, and they will activate immune system to destroy it so fast that there will be no symptoms of disease. You won't get sick again. T cells now. What happens with T cells after they mature and activated? They differentiate into three types of cells. Memory cells, we said about them. Also, those would be cytotoxic or killer T cells and helper T cells. Well, cytotoxic or killer T cells will be killing cells, destroying them by lysis. It means making a hole in them and the cell dies. Also, cytoplasm goes outside. Or by producing special chemicals called cytokines. What about helper T cells? They exist to activate, they help activating other B cells and cytotoxic T cells. Now, we already said about tolerance. Our lymphocytes should not respond to our own self-antigens. And what do we do with those lymphocytes that respond to self-antigens, that try to destroy our own cells? They will be either deleted or their activation will be prevented. Again, if either of those steps fails, then the person develops autoimmune disease, when our own immune system attacks some certain types of tissues, our own tissues. For example, if somebody has rheumatoid arthritis, it means that their own immune system attacks tissues in their joints and causes arthritis. Now let's talk in details how B cells work. So B cells, when they are activated, they produce antibodies, and they are effective against bacteria, viruses, and other types of pathogens, usually when they are outside of the cells. So antibodies fight pathogens that hasn't entered our cells yet. Another name for antibodies are immunoglobulins. You need to know both names, or IG. There are 5 main classes of immunoglobulins: IgG, M, A, E, and D. And each of them is made of 2 regions. One of them is a constant region, which is identical for all antibodies. The top part is variable region, which will be different for different types of antibodies. How they work very shortly. They approach pathogens, viruses for example, and attach themselves to those viruses. Clump them together, so right now viruses cannot do anything already, but they still should be destroyed. For that, antibodies together with viruses are taken inside of a big macrophage, and macrophage will destroy them. Those 5 classes of antibodies or immunoglobulins are similar, but they have slightly different structure and their functions can be slightly different. For example, immunoglobulin A usually is found in mucus, saliva, tears, and breast milk, and it protects against pathogens like all others. Immunoglobulin D activates beta basophils and mast cells. Immunoglobulin E protects against parasitic worms and takes part in allergic reactions. Immunoglobulin G is secreted in the blood by plasma cells, and also it's capable to cross the placenta into the fetus. And finally, immunoglobulin M has the most complex structure, and it's responsible for early stages of the immune response. Actions of antibodies. A few more details about how they work. So you already know that they inactivate the antigen by binding to it. Antibody binds to antigen. Right now, antigen cannot move freely anymore. Next one, several antibodies bind several antigens together. Now they are even more immobilized. But that's not it. That complex antibody-antigen can activate complement cascades, those complement proteins that cause inflammation, chemotaxis, and lysis, all those mechanisms that we talked about in Immune System 1 chapter. So basophil, for example, when antibody with antigen is attached to basophil or mast cell, it starts producing chemicals causing inflammation. That complex antigen-antibody also can promote macrophages to approach and take them inside for destruction. So those would be actions of antibodies. Now let's talk about T cells. Cell-mediated immunity. So, while B cells work with pathogens sitting outside of the cells, between the cells, in the blood and other body fluids, T cells try to destroy our pathogens that are already invaded our cells, that are intracellular microorganisms, viruses, fungi, bacteria, parasites that are inside of our cells. To kill those bacteria or viruses, our cytotoxic T cells have two options. First, they lyse virus-infected cells or tumor cells with help of a special protein called perforin. That perforin makes a hole in the plasma membrane of the cell. All that fluid out of the cell, organelles, go out and the cell dies. Viruses die together with the cell. And also, cytotoxic T cells can produce cytokines, special chemicals that will attract phagocytes and cause inflammation. As well as it's true for B cells, T cells can produce memory cells also for future encounter with the same type of infection. And that is immunity. So, when we encounter the same antigen again, we don't get sick thanks to the immunity. You need to know 4 types of immunity. Active natural immunity. Natural exposure to an antigen. You get chickenpox or flu. Excuse me. You get sick. You recover, and you will never have chickenpox again, or mumps or measles. You can have flu again, but the same type of flu virus will not cause any disease for at least 1 or 2 years. After that, flu virus just change and it can cause disease again. Active artificial immunity is vaccination. So, when they introduce vaccine to your body, they expose you to an antigen, usually weakened or dead antigen, and your immune system produces antibodies, memory cells, and so on. That is active immunity because it activates your immune system to work. Passive natural immunity, it's when you receive antibodies produced by somebody else. That can be your own mom if you are a baby. So mother's milk can contain antibodies produced by mother's immune system, and she can transfer those antibodies to her fetus or later to her baby when she's nursing the baby. And that's why it's so important to breastfeed the baby, because mother's antibodies protect baby from infections for at least 6-7 months. Passive artificial immunity. It's when one person, usually they use animals, produces antibodies and they are transferred to a non-immune animal or a human. That is called passive artificial immunity. To learn more about immunity, types of it, and more details, please watch the animation under the video. There will be a link. Remember to answer the questions to Chapter 21, Immune System, Part 2.